biomedical sciences is really revolving around trying to understand how individual cells go awry. So at the core of this are stem cells, originally, developmentally. These are the primitive cells that have the capacity to give rise to all the cells of an organ. And I'm going to spend the next bit of this talk, now that you have a good grasp on cells in general, I'm going to move over and talk about stem cells per se and how they, and the understanding of stem cells has really turned out to be quite a revolution in our understanding of biology. So first of all, the terminology, stem cells. The definition of a stem cell really comes into two parts. A stem cell is a cell, and now you know what cells are, that can self-renew. That means it can give rise to itself, make a copy of itself, divide and make two copies of itself. But a stem cell doesn't just give rise to itself. It has the capacity to divide and make two of itself, and it can also give rise to a more mature cell. So a stem cell is a cell that can keep dividing, but one of its daughters can become a mature cell, like a brain cell or a muscle cell or a heart cell. So that's all there is to a stem cell. The difference is the term that comes before it. So we talk of pluripotent cells. That means this is the most potent, the most powerful cell. Stem cell is called a pluripotent cell. This cell can give rise to every single cell of the body. So it's a single cell that can divide and give rise to itself, but it can give rise to heart, brain, liver, every cell of the body. And I'm going to talk about these cells, these particular cells, a little bit more. They're derived from the embryo, from an embryonic, as an embryonic state. Once the body begins to be formed, organs begin to be formed, and these pluripotent cells presumably disappear. They mature. They become more mature. But quite amazingly, every organ of your body retains a primitive cell that can give rise to cells of that organ. So now we know that heart has stem cells that are generally quiescent, generally sleeping, but periodically can be revived to divide and give rise to heart cells. This is true for skin. As we know, when we have a cut, we have repair. We're left with a scar. It doesn't always form exactly correctly. Brain, liver, all organs have a small amount of these stem cells. And our view of this now is that these stem cells, and I'll talk about this again a little bit more later, but in generally across all organs, these more mature organ-specific stem cells are called multipotent stem cells, because a liver stem cell can't become a heart cell or a brain cell, but it can become all the different cells of the liver. And a brain cell can give rise to all the cells of the brain, but it can't become a liver. So these are more restricted, and they're multipotent. And we believe actually that throughout life, small damages may occur to your organs, and there is a micro-repair system that's underway all the time that helps us out as we age and doing micro-repair. And I'll repeat this again later on, but part of the challenge of modern biology is to tap into these existing micro amount of repair and try to amplify that so that larger amounts of repair can occur from the small residual population of cells that remain. But if I move back now to the pluripotent cell and spend a little bit more time talking about these cells and their remarkable capacity and how we can actually harness the use of these cells and how we are harnessing the use of these cells in biology. So these are cells that you can grow in a plastic dish in an incubator in the right tissues, and they alone can give rise to muscle, and a single cell can give rise to these different cell types. I want to show you a movie. Now this is of a small worm called a C. elegans, but this is the first cell division of a cell. So this is just two cells. This is right after the first cell division. But this gives you an idea of what, this is sped up. So I couldn't do this for humans, or we'd be here all night waiting for this to continue. But this organism is dividing, and you'll see in a moment that it changes from just cells dividing to the beginnings of making structure. So you're beginning to see the formation of the structures of the worm. And it's on the final stages now. And this is now made up of several thousands of cells. So from a single cell, this develops into a fully grown, mature C. elegans worm that can function and move around and live and reproduce. And this took 14 hours and 46 minutes for this to occur in real time. So single cells, single stem cells, have the capacity for making entire organisms. Imagine the instructions that are going on, the communications that are going on between these cells to inform each other where they need to go at any one particular time. These are human embryos at an early stage. This is day one of a fertilized egg and sperm. So this is right after the egg and the sperm have joined together. And day two is the first cell division. At still going on to day two, we have the beginnings of what's called a morula, where at this stage, all of these cells that you're seeing at each of these stages are equally potent. They're all, at this stage, they're still all pluripotent. If you were to take one of those cells out, that cell could grow and give rise to all the other cells of the body. But as these cells mature, they differentiate, and particularly on day 5, you can see that there's a circular membrane surrounding a mass of cells in the middle. And this is a critical stage of development where the placenta is forming, but the cell mass that becomes the whole organism is in the core of the blastocyst. And the majority of human embryonic stem cells that are available for research now are derived through in vitro fertilization, which was pioneering work was done here in Melbourne by Australian scientists that contributed greatly to this methodology of generating IVF. But the advantage here is that you can, many of these thousands of these fertilized eggs are frozen down, and individual of these cells, fertilized cells, can be harvested, and the active inner cell mass can be placed in a dish, and they can then be grown up indefinitely under certain conditions. So these cells, these pluripotent cells, then can be grown, and those under certain conditions can give rise to all the different cells of the body, and it's quite extraordinary in a dish. This whole process can be done in a dish. So as I said, many cells, many diseases are really diseases of cells. Alzheimer's disease is diseases of specific cells in the cortex and in the hippocampus. Heart failure, of course, is a result of diseases of the heart. Parkinson's disease is a very specific disease. It's not a general disease of all cells, but uniquely targeted cells which affect motor behavior in humans. And we can go through the list of diseases, all of which really correspond to diseases of very specific cell types. So it behooves us, it is the challenge for us to understand the nature of these diseases. Now, how can human embryonic cells help us along these lines? Well, first of all, they're human. So they allow us to study disease in the context of humans. They're self-renewing, so under certain conditions they can be propagated or grown indefinitely. They can be frozen down and shipped around all over the world, so different experimenters can use and do the same experiments. They also are used practically for a variety of issues. One is to understand the basic principles of how early development occurs in humans, which is something we have not really had experimental access to prior to the development. We, of course, envision the idea that these cells, if they're turned into a particular cell type, like a heart cell or a brain cell, they can be transplanted and replace those cells that have been damaged. That's the promise, to some extent, of these cells, but it's only one of the promises. The other one that I'll talk a little bit about is that human cells, these human cells can be induced to become very specific cell types and used to model human diseases in ways that we've not had before. They can also be used to test drugs and I'll talk a little bit about that in the future, but another feature is that our hope, more than hope, is that, and the plan is that we'll be able to build artificial organs from these ES cells because of their capacity to give rise to all tissues. So the process of cell therapy looks something like this. There's the sperm and the egg are fertilized, the morula begins, the inner mass is harvested and put in culture, and then those cells can be differentiated into all lineages. And here's just an example of a condition where cells had been induced, embryonic cells have been induced to give rise to heart cells. And here you can see that they've organized themselves into a structure where they spontaneously beat. So this is the beginnings of what we hope will be organogenesis, or at least parts of organs can be generated in culture. I should also tell you that this is a wonderful field of investigation that is really merging between biologists, medical scientists, engineers, mathematicians. It's cross-disciplinary. It's an extremely exciting area of confluence of ideas. So the view then, of course, with cell therapy is that these cells would be differentiated into the specific cell type and transplanted. There are many, honestly, there are many hurdles yet to be overcome, and that is to make sure that these cells become exactly the cell that you want them to become, and that they all differentiate, that is, they all become mature and don't retain this capacity to divide. The concern for making tumors remains within this field at this point, but progress is being made. The other problem with the transplantation aspects of this is immunity. So we're making the embryonic stem cells from an individual that has a specific immune system, and the